Welcome back to Plantopia. Plantopia is the plant health podcast of the American Phytopathological Society. The views and opinions expressed by our guests are their own and do not necessarily reflect the views of the host, the American Phytopathological Society, or any affiliated entity. I'm your host, Matt Kasson. I'm an associate professor of forest pathology and mycology at West Virginia University. This is the 11th episode of Season 5, and today I'm excited to welcome Dr. Lina Quesada, the William Neal Reynolds Distinguished Professor and Vegetable Pathologist in the Department of Entomology and Plant Pathology at NC State University in Raleigh, North Carolina. Dr. Quesada holds a BS in Microbiology and a BS in Biology from Universidad de los Andes in Bogota, Colombia. She also holds a PhD in Plant Pathology from Michigan State University, East Lansing, Michigan. Dr. Quesada was a USDA NIFA postdoc scholar at Michigan State University before joining the faculty of NC State in 2013 as an assistant professor. She was promoted to associate professor in 2018 and full professor in 2022. She has served in her current role as distinguished professor since 2024. Dr. Quesada is among the world's experts on oomycete pathogens causing downy mildew diseases and sweet potato pathogens, including fungal diseases and diseases caused by plant parasitic nematodes. Given her reputation for excellence and her collaborative nature, Dr. Quesada's research program has generated over $50 million in awards as PI and co-PI on diverse federal and international grants, including funding from the Bill and Melinda Gates Foundation to advance sweet potato production in Africa. Dr. Quesada is co-author on some 100 peer-reviewed research articles and has mentored 14 grad students, 10 postdocs, 8 visiting scholars, and some 60 undergraduates during her career at NC State. She has also co-authored hundreds of extension publications in both English and Spanish. She has been an active member of APS throughout her career and has served as chair of the APS Leadership Institute, as well as the APS Post-Harvest Committee. She is the recipient of the APS Syngenta Award in 2023, the APS Schroth Faces of the Future in Mycology Early Career Award, and the APS Hewitt Award in 2017. Dr. Quesada, it's such a pleasure to welcome you to Plantopia.
Thank you so much for the invitation, Matt. It's really nice to be here.
Looking over your CV, I see that you did an undergrad in Bogota, Colombia. Are you from Colombia?
Yes, and I am from Bogota, Colombia.
Okay. Anybody who knows anything about Bogota, it's that it's a giant city, right? It's certainly the largest city in Colombia, but it's, the third largest city in all of South America. And imagine to think about city life and ending up a vegetable pathologist. I wanted to connect those dots a little bit and figure out how you first got interested in microbiology, which was one of your undergraduate degrees, and when you first got really interested in plant pathology.
Oh, that's a good question. So I became exposed a little bit to agriculture actually through my dad. He worked in a bank where he had to interact a lot with large agricultural producers. And he would take me sometimes to those visits. So I got to visit, for example, like really big like rose plantations or like banana plantations, coffee plantations, other types of ag. But I was always fascinated by that. And then he actually ended up buying a small farm, maybe like an hour and a half away from Bogota. And we would go there over the weekends and we would grow citrus and a few other like tropical type fruits. So I became a little bit interested in that. But that said, when I went to college, my dream was actually to be an immunologist and like basically develop vaccines against the most horrible diseases I could possibly think of. My mom was zero excited about that career goal. So then I ended up getting a job in a lab at the university where I had to like inject a bunch of baby mice and things like that. And I quickly realized that was not the path for me. And I was actually a little bit lost there for a while. And I ended up taking a lot of classes in both biology and microbiology, which is why I ended up doing 2 majors. But then I took a class on basically plant immunology. And that was like my aha moment of this is what I want to be, a plant immunologist, aka a plant pathologist.
So the small farm your dad purchased, that was before you started college?
Oh, yeah, that was when I was a kid. So I was always excited about nature. I felt like I always wanted to do something biology. I just wasn't sure exactly what.
And did you feel like there was a lot of overlap in the two, like doing both a bachelor's in biology and microbiology? Which one did you like more? And was it just easy to do both because there was a lot of overlapping curriculum?
That's a really good question. So they actually don't overlap as much as what you would think, because microbiology was really more like the traditional microbiology that you would take here. A lot of micro classes, food microbiology, medical microbiology, virology, bacteriology, all those things. Whereas the biology major was more oriented towards ecology.
Yeah.
And that, to be honest, was less exciting to me. I did take some really nice classes about plant biology, plant physiology, things that became more relevant to really becoming a plant pathologist. But I think definitely I was more a microbiologist at heart. I did had a small phase that I thought about being an entomologist. I even got like a ladybug tattoo and everything. I was very committed. But then again, as I started taking like plant microbe interaction classes, it just became so obvious that microbes are like way cooler.
Yeah.
So I
Well,
went for that.
and of course, we can't discount insects as being important vectors for plant diseases and viruses and
Yep.
things like that. So it all connects. So in Colombia, I'm just thinking about like the main crops there: coffee, avocado, like palm oil, sugarcane. So you said you visited some of those coffee plantations and that when you were with your dad going on these
Yep.
visits to these different agricultural regions. Clearly different crops than what you deal with today, but as we'll get to later on here, they're all connected because some of these pathogens are certainly have a broad host range. Now, I saw that you were a visiting scholar at The Ohio State University in 2006, and this is before you started your PhD at Michigan State. How did you end up there, and what did you work on?
Yeah, it's another good question. At the moment, I was working in Colombia in CIAT and a little bit also in the University of Los Andes, working with like tropical fruits, Phytophthora infestans, and also some bacterial diseases of cassava. And I think through those experiences, I realized that I really wanted to do science that really applied to growers. And at that time in my country, there was a lot of social unrest that it was not really realistic for me to probably have that career path. So I decided that it was probably time to maybe think about getting like an advanced degree overseas. And I started just sending emails to explore possibilities. And I was really lucky that Dr. Sophie Kamoun, who was back then in the Ohio State University, replied back to me and said, "Hey, I have a visiting scholar position. Would you like to come to the lab for, I don't know, maybe a year or so?" And of course, I said yes. And I was with him for a while working on Phytophthora infestans plant microbe interactions and with Dr. Jerome Boss, who is now in the James Hutton Institute. And that was a fantastic experience, but it also exposed me to extension.
Yeah.
So the lab next door to Sofian's lab back then was the lab of Dr. Sally Miller, who was a vegetable pathologist. So I befriended a lot of her lab members and I learned about extension and I thought it was like the best thing ever because we don't have extension in my country. And again, through all those emails that I had sent, I got really lucky that Dr. Mary Housepick of Michigan State emailed me back and said, "Hey, I'm actually looking for a PhD student. Would you like to maybe talk?" And since I was in Ohio, I could actually go and talk to her, which was great. So we
Yeah.
connected and I ended up applying and I accepted a PhD position with her. And I'm still really in touch with Sophie. The training I got with him is something I still apply, but I think my heart was really on like doing more extension and trying to work more directly with growers and producers. So I was really lucky.
And just thinking about good mentors is like those are relationships we develop and retain for life.
Yes.
And as much as we talk to the people that kind of shaped our early career, I know that probably a number of the alumni from your own lab that have gone on to industry and was looking through your CV and seeing so many of them ended up in industry. And you still maintain those contacts professionally, but also because you care about the development of your mentees through the rest of their career.
Oh, absolutely. I think that it's not always the case, of course, because it has to be like a mutual decision. But I
Yeah.
definitely have always that door open that if people want to continue to have that career long relationship, like the door is always open. And I'm lucky that really a lot of my previous lab members, we are still very much in touch. And I get to work actively with many of them because many of them ended up staying here in North Carolina because they went to industry. That has been very enjoyable.
So I want to just come back to the visiting scholar aspect is that I noticed that you yourself have had a number of visiting scholars in your own lab. You see that like it's a deliberate thing to bring in visiting scholars so you can do the same thing for them.
Oh, absolutely. I think the experience that I had with Sophie, and I often tell him that was like absolutely transformative for me because it wasn't only just going to work with an excellent scientist and something that I really was interested in, but I think also was a moment for me to build up my confidence Colombia can be hard for a female scientist. I think the US offers a lot more equal opportunities for female scientists, which I think I didn't realize, I think, how different that it was until I moved here. And for me to be in Sofian's lab and be given the opportunity to really participate, to be a valued lab member, to just have the freedom to really learn without those things getting in the way, it was incredible. And like you said, I wanted to help maybe other Colombian students or from elsewhere to have that opportunity to pursue their dreams. Yeah, I have tried to do some of that when possible.
Now, one of the other things that you mentioned was you became aware that extension existed, and that's something that didn't exist in your home country. And looking over like your own body of work, your research productivity is just really impressive when I look around it. And one of the things that caught my eye was you at least didn't list a lot of formal classroom instruction. I wondered what your actually appointment breakdown was at NC State. I assume it's mostly research and extension. What's the breakdown there?
It's actually 50-50.
So 50 research, 50 extension?
Yes, and I actually chose that. So in NC State, we do the effort a little bit different, that you can choose a little bit in your kind of like the framework that you're evaluated and what's your split gonna be within the realm that you were hired. And I honestly purposely chose 50-50 because that's how I feel. I feel that every piece of research that I do in my lab should serve an extension need. And it doesn't matter if the research is very applied in the sense that it could be doing field trials or things like that, or if it's very molecular, using some of the tools that I learned more with Robin Buell or with Sophie and Camoon. All of it is in service to North Carolina agriculture. So I do really feel that I'm like half and half.
Yeah. It's funny because if you talk to a lot of academics, you'll hear them say things like that, but it's clear that you have a strong body of evidence that supports that. Like you're really interested in basic science and you do really excellent basic science, but it's so important for you to take it that next step and connect it, help the grower understand why you have to sequence a genome of a downy mildew pathogen, because without it, you can't understand the interaction with the plant and breeding for resistance and things. I think you go above and beyond. And part of that is that's my job, but you're really good at it.
Oh, thank you. And I think you captured it really well. Like, I know it's unusual to look at my CV and see somebody so committed to extension because you're right. If you look at my CV, some of it reads very molecular, very basic science. But I think my goal always begins and ends with the growers. To me, it's just more sometimes I need to use field tools and sometimes I need to use bioinformatic tools. But that is really important to me, like that my research addresses real, tangible agricultural issues. And you said it, that I can really take those solutions to the hands of my growers. That is my absolute priority. Publishing is very important. And I'm a huge fan of publishing in the sense that I think it's the mechanism that we need to use to document the knowledge that we're advancing. But you will probably also notice that I mostly only publish in APS journals. I have a purpose decision that I publish in my society
That,
journals. A lot of people also ask me like, why aren't you publishing? And I don't know, BNC Genomics, Nature Plants. I don't want to. I'm not doing my job to get highly cited, glamorous papers. It's really just a mechanism for information dissemination, and I much rather give my citations to my society.
yeah, I'm in agreement on that. And I really am actively involved with APS and MSA. And I feel like if you really stand behind your societies, you should support their journals.
Absolutely. And in the end, the best reviewers of your work are going to come from your society. And the most captive readers of your work that are really going to use what you're doing far more immediately are the people in your society. So that's the community that I'm trying to impact.
Sure. Now, you're among the world's experts on downy mildews, and these are obligate oomycete parasites of numerous plants. I'm sure many of our listeners will be familiar with downy mildews. Of course, we've had a number of people talk about them. But in the interest of ensuring that all our new listeners and maybe listeners that are part of the general public but not experts in plant pathology can follow along, what are the defining features of a downy mildew disease? And how do they differ from something else, other mildews like a powdery mildew?
Oh, that's a hard question visual, but I'll try to answer it. So, organismically, downy mildew disease is caused by an oomycete type pathogen, which is like a cousin of an algae. They're very different from true fungi. They're diploid and they have different cell walls, a number of other features. But powdery mildew is really caused by true fungi. As far as visual features, that is hard because we really have downy mildews that can look white. The sporulation in the leaves can look white, more similar to a powdery mildew. And then we have downy mildews that have dark sporulation. What are you seeing? Cucurbit downy mildew, basil downy mildew. So I do think that visually could be tricky to tell which one it is.
But more speaking from the symptom side, typical, like what's an iconic symptom of a downy mildew disease?
That's a good point. So I do think that downy mildews obviously tend to be foliar, just like powdery. And because they're obligate, the lesion that they form in the leaves tends to be more delimited by the veins of the leaves. If you have a very strikingly angular leaf as far as their veins, like cucumber, the lesions look a lot more sharp in their edges, right?
Yeah.
If you see a basil leaf, it really looks almost like rectangles because those sections of those basil leaves look like that. And I really think you're right, that's pretty different from the powdery mildews. The powdery the lesion tends to just really be more like a circle, the white sporulation, regardless of whatever it is in the leaf. So that would be different, yes.
And then on the downy mildews, remind us, do you get more sporulation on the underside of the leaf?
Underside of the leaf, but there are some downy mildews that also grow a little bit in the top. I have seen that with some, which is really interesting.
Yeah. And I think it comes back to this idea that like we have terms like mildews that could represent true fungi as well as oomycetes, which are fungus-like organisms. They're eukaryotes, but they're not fungi. They're not true fungi. But for so long, they've been studied alongside fungi because they're very fungal in their habits. Or we could say fungi are oomycetes in their habits, however you want to frame that. So downy mildews can impact a number of hosts, crucifers like cabbage and mustards, grapes, of course, grape downy mildew, and numerous vegetables. And that's where it intersects with you because you're a vegetable pathologist. You've studied them on numerous hosts, hops, cucumbers, or cucurbits, spinach. What crops do you think, generally speaking, are most impacted by downy mildews at the moment? And really coming back to something we're already starting to talk about is how has your work helped address growers' needs?
Good question. So as far as like crops impact them, there's so many, honestly. But I think that have been economically significant here in the US. Cucurbit downy mildew is really the number one because it really became a barrier to cucurbit production in the eastern part of the US. Hop downy mildew too, I think it's just that when the hop industry used to be in the east coast of the US, it was very impacted by both downy and powdery mildew. Once it moved to the west coast where it was drier, that kind of took care a little bit of the downy mildew issue. Not that it's non-existent, but it's much more manageable, I think, than other diseases that they have. A few years ago, we also had a big problem with basil downy mildew, especially because basil can be produced in greenhouse, which can get fairly humid. And this is a disease that can appear in transplants. If you would buy a little basil plant out of Home Depot, likely it would have downy mildew. That's where we got our inoculum for a couple of years.
Oh, wow.
So until that got under control, it was just very ubiquitous. And something like that also happened with impatiens downy mildew a few years ago. And again, totally different industry ornamentals, but impatiens is such a loved plant by many because they're just beautiful. And suddenly here comes this downy mildew and really devastated for a few years until the seed companies for impatiens really caught up with some tolerant varieties. And then fungicide interventions, of course, helped there for a while.
Could you clarify something for me?
Yeah.
The downy mildew pathogens, like the one you study on hops and the one you study on cucurbits, are they native pathogens?
That's a good question. So Pseudopronospora cubensis was first found in Cuba. There's always this belief that a lot of these pathogens really comes from where like the host center of origin or centers of diversity are.
Sure.
Cucurbits have a few different centers of diversity. There's sort of like one focal in Africa, one in Asia, one in Australia. I don't think that we really know exactly where P. cubensis came from. We just know that first description happened in Cuba. With Pseudopronospora humuli, same thing. So I believe, if I recall that literature, there was a strong belief that it may have come from Asia. There's a wild hop in Asia that very tends to have the pathogen present. And I believe some of the material that was many moons ago moved here into the Americas came from some of those areas that start breeding programs.
Just coming back to this, what you mentioned is that you said When we were growing hops in the East, we had problems with both powdery and downy mildews. And of course, we know with cucurbits, downy and powdery mildews are a problem. Are downy mildews more of an issue than powdery mildews on cucurbits, generally speaking, in the garden? Because I see a lot of powdery mildew on my cucurbits, but I see a lot of downy mildew on my wild grape around my yard, but I don't necessarily see a lot of downy mildews in my garden.
Yeah, that's a really good question. And it ties to the question you asked me a little bit ago, which is how is your work impacting the grower? So
Yeah,
here's an answer to join those two together.
perfect.
Yeah, I think the issue is that it depends on the cucurbit. And that is something that we didn't understand until a few years ago, because we will observe exactly what you're describing, that there could be one cucurbit that had a heavy infection of downy mildew, and it would be right next to another cucurbit, and the other cucurbit was okay.
Mhm.
So my lab did a lot of research that ended up discovering that what we call Pseudopronospora cubensis is actually comprised of two clades. And this was initially proposed by Dr. Marco Tynes in Europe. It was a phylogenetic study he did in Europe, but he only included one strain from the US.
Um,
But he proposed a clade classification. However, his sampling, because of the nature of the study they were doing, was opportunistic. So he couldn't quite clarify if there was like a host division between the clades. And that's something that my lab was able to really completely clarify, that clade 1
uh-huh.
tends to preferentially infect squash, pumpkin, and watermelon, whereas clade 2 tends to preferentially infect cucumber and cantaloupe. For the growers, this was really important because many times I would have, let's say, watermelon growers telling me, like, we're not really getting down in mildew. We struggle more with things like gummy stem blight, wilt, and anthracnose. So for them, downy mildew was not a big issue. Same with some of my pumpkin growers that would say, I hear you about downy mildew in cucumbers. For us, powdery mildew is really a big issue. But I would talk to my cucumber growers and they would be very concerned because they would be drowning in cucumber downy mildew and with not a lot of great tools. The fungicides that would work really well for my watermelon and my squash growers for downy mildew were not really performing at the same level for my cucumber growers. So fast forward, we ended up discovering that it is because the clades not only have host preference, but they also have differences in fungicide sensitivity.
Oh, wow.
So basically, it's important for you to know the clade first because the cloud of spores that is coming from right every year may infect one host and not the other, but also depending on the host that you're talking about, a fungicide may or may not work. So we're really trying to inform our management recommendations basically with like pathogen population genetics so that we can first make sure that the growers have better management outcomes for the investment that they're putting in sprays or whatever. But second, that we're protecting the fungicides from fungicide resistance because
Right.
clade 2 just tends to develop fungicide resistor much faster. And that's not a good situation for anybody that likes pickles in their burgers.
Well, that's really interesting. And thank you. That was really clarifying. It explains some of the disease dynamics I think the average gardener might experience in their own small little plot. We think about how this affects big growers and commercial producers, but it has utility for that person who only has a raised bed garden and they're growing like 4 crops right next to each other. So let's talk sweet potatoes a little bit.
Yeah.
After all, this is the season for feasting on sweet potatoes. You've studied numerous diseases of sweet potato, including those caused by fungi, nematodes, etc. Black rot caused by Ceratocystis fimbriata, the scurf caused by another fungus, sweet potato root knot nematode, rhizopus soft rot, geotrichum sour rot, fusarium root rot. Even if you haven't worked on all these, you've definitely published fact sheets for these through the extension arm of your university. And thank you for that because they're wonderful resources. So based on your publication record, I'm inclined to say that black rot caused by Ceratocystis fimbriata poses the greatest contemporary threat to sweet potato production. Is that fair?
That would have been for a few years ago.
Okay.
That-
Okay.
that- Now we have it fairly under control. But yes, that was probably the first extension emergency that I had to deal with when I started here was a big outbreak that we had of black rot in 2015.
Okay. You said a couple years ago. So what's posing more of a risk? The nematodes, or is it something else? Or
I would say the nematode definitely, unfortunately. So we had the issue with black rot in 2015, and then right after that in 2016, I remember still I was contacted by an extension agent that I really liked, and he said, "Lina, I'm really concerned. I'm seeing all this nematode damage in sweet potato. And I am not a trained nematologist, unfortunately. When I was in grad school, the nematologist that was at my university had retired. So my nematology training was a Wednesday morning in grad school. That really concerned me because I'm like, I don't have the training, and our nematologist at NC State had just retired. But it was sweet potatoes. I felt that at least I needed to try to help, even though it wasn't really under my purview. So I went to see this field, and it did look really bad. And we decided to see if we could put out a field trial. And we ended up finding out that the nematode in question was Meloidogynon turlobi, which is a very aggressive root knot nematode that is found in like Cuba and some parts in Florida. At the moment, it wasn't recognized to be in many areas of the US because we were just not looking for it. I did trials for many years. We came up with some stopgap chemical recommendations, but I also really advocated actually to get a new nematologist with the industry. And we were really lucky that we were able to hire Dr. Adrienne Gorney in 2019. So she's now our nematologist at NC State, and she really took over that issue. We partnered with Clemson through an SCRI project, and Adrian is part of another SCRI project led out of NC State. And through those two projects, really for several years, we learned a lot about this nematode. It has a really broad host range, which is not good news. It's crop rotation then of limited value. We have some chemistries that work, but they're very expensive. And we're trying to just mostly accelerate breeding efforts. And we have a very good sweet potato breeding program here at NC State led by Dr. Craig Yancho. And they have a couple of varieties that look promising. So that's where we're at, trying to see if we can use chemical recommendations to short term keep that at bay until the breeding program comes with a more permanent solution.
Okay. I want to rewind back to when you first came upon these fields that were heavily infested with root knot. You said the fields looked bad. Let's just remind our listeners, generally, what kind of above ground symptoms are we seeing? Or was this more you were seeing in the harvested sweet potatoes themselves? Tell us a little bit about that.
Oh, it's a good question. A little bit of both. So you would definitely see some reduced stand in some of the sweet potato fields. Sweet potato fields tend to have a pretty, well, at least the rows, very dense canopy. It's a really beautiful plant. It's one of those plants that once you put it out, it doesn't get a lot of diseases during that growing cycle other than the nematode. Like we don't really spray fungicides the months that the plants are growing. It's really only at planting and post-harvest. But you would see that the row would become spotty. There would just be like a plant that died early on or something like that. So you definitely see that. And then you said it correctly, once we harvest it, is that you would really see it. Because you would see these roots completely cover in galls.
Yeah.
It was not 3 galls, it was like 50 galls.
Yeah.
Gall on top of gall. It was just very striking. I really had not seen something like that before.
And how does that impact the overall like tuber production? Is it just like they're really deformed, or they're just like they're way less, or?
Both. You definitely have way less, but they're also just not marketable. Like they're even for processing, I don't think it's something you could really easily use. So yeah, the effect was pretty devastating. And again, now we have really good chemical recommendations, so the growers have adapted to that new reality, kind of like accounting for those extra input costs.
Yeah.
I think it has been hard, and they would love to move away from that as soon as possible. There's
Yeah.
a resistant variety, but it's how we're coping now.
So I want to come back to a point you made casually, but it's one that I think it's worth explaining to the listeners, especially those that may not have a background in plant pathology, is that you said we don't spray a lot and we don't really have to manage a lot during the active growing season for sweet potato, because a number of the diseases you've been working on are actually post-harvest diseases. So these are diseases, the inoculum is already there and they may already be infected when they go into storage. And then over time in storage, these rots, whether they're the soft rot or a sour rot or a black rot, they develop, rendering the sweet potatoes unusable in storage, correct?
You said it perfectly. That's really how it was. Because I remember when I was hired in this job, several of my sweet potato colleagues more on the production side told me, like, you know what? We don't really have disease issues in sweet potatoes. You may just be working in the other vegetable crops. And I was like, oh, okay. But I thought about it and I was like, I feel like I need to check this on my own. So I went to the packing houses. I love going to sweet potato packing houses. That will never get old. It's so fun. one of my favorite stops in the sweet potato packing houses is the coal pile. So when sweet potatoes get harvested, they get cured and stored for up to a year. And then the packers slowly take kind of bins out to pack and sell either domestically or an export. So when those boxes come out, they get rinsed. And then there's like a line of pickers that are basically picking out all the roots that are like diseased, broken, whatever, right? And only
You're right.
pretty roots continue the packing line process. So that coal pile is like my gold mine, right?
Yeah.
If I would go to that coal pile and just by eye, you could see, I don't know, 6 different diseases, right?
Yeah.
So I would take samples to get a sense of what are the sweet potato pathogens. Because another thing that I think is tricky and it wasn't obvious to me when I took this role is if you compared in the history of plant pathology, the number of sweet potato pathologists that we have had versus the number of, let's say, tomato pathologists that we have had, I assure you that the difference is striking.
Yeah.
We just don't have the sweet potato pathology expertise that we have in other crops. We are easily 20 years behind every other crop that I work with. So it really has been starting that knowledge from the ground up of like understanding, okay, what are the diseases that we have, which, as you said, are really truly completely post-harvest, but which ones are actually starting in the field. We are not recognizing this. And then we're seeing them post-harvest. And then what interventions can we do early in the growing season to prevent that they appear? And some of those diseases are black rot and geotrichum sour rot, which to me, that is probably the number one issue that we're working on right now. And also macrophomina. But
So you were saying that those are specifically start in the field.
yes.
So let's stick with black rot since you've worked with it so much and published on it. It may not be a problem because once you tackle something, you help to provide solutions to solve it. But I find it interesting that you're from Colombia and that coffee and cacao are both also hosts for Ceratocystis fimbriata. And this kind of brings up what I think is an interesting topic. And it's that what we do to better understand a specific pathogen plant interaction could often be applied more broadly to understand other parallel like pathosystems. Have you found that what you've learned in this system can then be applied towards the crops in your own home country? And did that foster any collaborations in that sense?
It's a really good question. And yeah, I mean, I actually saw it a little bit the other one. I'll tell you in a second why. But no, it unfortunately has not really fostered collaborations in that way, mostly because it really is sad to say, but Colombia doesn't have a good infrastructure to fund a lot of research. And as same thing happens from the US, like you cannot really easily fund other countries to do research grants that we obtain here.
Sure.
So that has always been a barrier. But I find it so interesting that you notice about the host range because I think I noticed that immediately when the emergency popped up. I didn't know much about that pathogen. So I went to the USDA host fungus database. That's usually my first stop when I'm doing something that I don't know. And I noticed the same thing. I'm like, oh my gosh, this pathogen goes to sweet potato, lettuce, and trees, and to that tropical trees. That is so interesting. And we just didn't know much about it, right? And something that I kind of use to your point of how does that transfer of knowledge look like is this pathogen is vectored by insects in other hosts. And when the black rot thing happened, the way we really were able to stop it was with a fungicide. We found an effective fungicide, got an emergency label for it, and we put the fire out. But I really wanted to also develop like mechanisms long term for people to manage it. And something that I noticed when I visited the packing houses when the epidemic was in full swing is that there were all these fruit flies.
Yeah.
It was so bad. It's like I couldn't talk to the packer and open my mouth because like flies were going to my mouth. It was really unreal. And I always wondered, could it be that these things are actually not just being attracted to it, but moving it around? And we started that research a few years ago with Dr. Anderseth and Dr. Matt Bertone, who are entomologists here in the department.
Mhm.
And we did. Kelly Avila was able to confirm that this fruit fly is able to transmit Ceratocystis fimbriata and sweet potato. And that idea came because I think this happens in mango. It's with a different insect. I think it's like a borer beetle.
Yeah.
If I remember. But I basically was like, oh, I bet this can happen with a different insect. And that's when I started pursuing that. So yeah, so the knowledge transfer definitely happens, just not in the direction you're thinking.
But it also speaks to something that like is really important. It's like you want to become as well-rounded of a plant pathologist as possible because you don't know when your research is going to intersect with some other system that you've never even thought about.
Yep.
While we're on the topic of sweet potato is in the morning glory family. And the morning glories, according to my research, are also susceptible to some of these diseases that impact or at least can serve as inoculum sources in terms of chlamydia spores for some of these diseases. Is eradication of like kind of morning glories, because I've seen them like act really weedy around like just rights of way and like along roadsides along farms. Says, oh, there's a big patch of morning glories. Is eradication of morning glories like any part of the strategy for controlling diseases of sweet potatoes?
Oh, absolutely. And I have to say that actually the sweet potato growers are exceptionally good at keeping their fields pretty clean of weeds. It's rare that I would see a lot of morning glories like in the middle of a production field. You would see them. You said it really well, like in the ditches around it. So you're right. They're definitely there and they can become an issue. And they are hosts for a lot of the pathogens. So I think what you're describing is completely possible. I don't know that it's, let's say, a bigger contribution that the inoculum we just have in the soil.
Yeah, no.
Because the morning glories are there, but I don't know how much they're supporting that side. Talking about the nematode, I think that could be one that would be a big issue, right? Because nematodes being obligates, they really are multiplying in that root system. So I could see how maybe the morning glory could make that situation worse.
Yeah. So morning glories are a host for the sweet potato root knot nematode.
I believe so.
Okay. While we're on the topic of disease of sweet potatoes, I wonder, is southern blight an important threat to sweet potato production, or have they bred for resistance and it's not an issue?
No, it is an issue. And again, it's one of those are like unrecognized issues when I did my coal bin visits. The
Yeah,
back
yeah.
houses. But that was one of the diseases that I noted. And I guess I saw it a little bit as low hanging fruit to try to fix it because, like the black rot issue, there are effective fungicides against southern blight out there. We just didn't have them labeled for sweet potato. And I don't know that people were recognizing that it was an issue. So we collaborated a lot with the IR4 project to do and to really with chemical companies too to screen a lot of those fungicides that we know to be effective for southern blight and other crops, but in sweet potato just to make sure that there wasn't like phytotoxicity or something like that. And we found several that are effective. We submitted those label requests through IR4. He actually already got a few labeled, and we have, I don't know, maybe 5 more that I think are moving through the pipeline of IR4. So we're very close to solving that one. And to your point of host resistance, so in North Carolina, we mostly grow this variety called Covington that came from the ancestor breeding program. It's an excellent variety. It's really pretty and yields really well, and it has what the breeders call a good disease resistance package. Things like fusarium are not a huge problem. Things like soulpox are not a huge problem. But black rot, southern blight, geotrichum, rhizopus, some fusarium, they are a problem. But in that case, then we try to rely a little bit on integrated pest management and do either cultural things or use fungicides to try to tackle that.
A recent grant of yours from the USDA FAS, I don't know if it's still ongoing. Actually, a couple of your recent grants were really on trade barriers for export of sweet potatoes. I don't know much about sweet potato production in general. I certainly don't know anything about sweet potato exports. How much sweet potatoes do we as a country actually export, and where are we sending them?
Oh my gosh, that is such a great question because I love talking about this. Like you, I also didn't know anything about this. Like sweet potato was a totally new crop for me. And it's really interesting because our growers are able to store the crop for up to a year. They can control a little bit the supply. So we had a very good domestic market, but I think North Carolina produces, I don't know, like 60-70% of the total US production. We're really just
Wow, that much, huh?
monster. We are a monster in sweet potato production. And I think at some point between the other states that also produce, we started saturating the domestic market. So some folks started to find export markets because sweet potato is such a healthy crop.
Yeah.
It really was welcomed, Europe, Asia, all these places. But the challenge was, how do we get this product to arrive there in good shape when they have to be in a port for a week? on the boat for another 3 weeks, then on the receiving port for another week. It was a huge issue. So that's something that we have been trying to do with fungicides, right? Like figure out, okay, what are our fungicide applications that we can use to keep the product looking really good. The European Union has very strict regulations for a lot of fungicides. So here, at least in my role, even though my role is really just to figure out disease management for sweet potato growers, I have extended that role to help them make sure that they can access export markets. So again, we collaborate with IR4, the Foreign Ag Service, the European Commission, which is like a pesticide regulatory entity in the European Union, to make sure that we're harmonizing the pesticide residue levels of pesticides that we are using in sweet potato so that the growers have access to those export markets. And that is the research that is being supported by those grants.
Now, some of the tools that these countries of import would use for detection, similar to what we might use like through APHIS, do you help develop some of those tools for detecting these pathogens levels, or is that not really like some of the stuff you do? Because I know we have all these modern molecular tools we can use to detect really low thresholds. Of course, you want to be exporting as clean a product as possible, but it's not like it's inoculum free. It's just below a certain threshold or asymptomatic or whatever. Have you worked on developing tools that are used in that kind of quarantine regulatory infrastructure?
So not really, because in sweet potato, the pathogens that I work with are not really under regulation for exports. And I forgot to mention that we do export about 60% of our acreage. And that is a very desirable market because it's 6 times the price that it is here. But none of the pathogens that I work with are like regulated for our importer market. So not so much that, no.
And you said it's mostly a European market where the goods are going?
Europe, Canada, some countries in the Caribbean, and some parts in Asia. Really, the limitation is the shipping time.
Sure, that makes sense.
I think that we could send to more places, but it's hard because of that shipping time. So
Now, you had another grant from USDA SCRI on harnessing genomic resources for disease resistance and management in cucurbit crops. Something we touched on a little earlier, but give you an opportunity to talk a little bit more about it is like, because this is like where your love and extension comes into play here. Can you explain to our listeners how one might leverage genomic resources to manage disease? I think that like drawing a line from A to Z is not always easy to do for members of the general public. Do you want to walk us through that, or?
yeah, no, that's a really good question. So I would say that probably a pretty obvious one that we have to mention, and you were asking about it, is diagnostics. I think robust diagnostics is something that we're always going to need in plant pathology. And our field has largely relied in things like ITS to do diagnostics. Now that we can sequence full genomes and basically understand what is like the pan genome of a pathogen species, we could probably find much better markers, not just for detection, but you said it to like quantifications. And sweet potato, we're not doing that, but if you think about waterborne pathogens, airborne pathogens, things that where the inoculum level matters.
Yeah.
What is a really powerful tool? And it's actually something we're doing in sweet potato. We're trying to develop a biosurveillance system for sweet potato packing houses. So the idea is to sequence a bunch of rhizopus genomes in collaboration with a few researchers that are working in that space, and then identify like a single copy nuclear gene that you could really use as a proxy to quantify spore levels. And that you can very quickly do with genomics, right? So diagnostics is an obvious one. Then think about things like fungicide resistance, right? Being able to diagnose management relevant traits, virulence, pathogenicity, fungicide resistance. That is something that we can also do better with genomics. Another thing that I'm excited about, and we're working in that space too, is host resistance. We know in plant pathology that at least some of the resistance in plants is mediated by NLR genes. Folks in Europe developed this technique called RenSeq, which is basically like an exome capture technique to specifically sequence the NLR family of genes. And that way you can essentially create like a catalog of like part of the immune system of a plant. Those genes are poorly captured by whole genomes. because those genes tend to happen in clusters of many genes that once you have the assembler, let's say, assemble your data, even if it's long read data, it may end up collapsing 20 genes into one gene. So you really lose information, even though genomic sequencing has come so far. So by using RenSeq, you might be able to capture a better catalog and then help breeders figure out, okay, are you finding in an area of a QTL that is strongly correlated with resistance to something? And then it may reduce the fishing expedition of which is the actual gene that is conferring resistance to something. So yeah, I think genomics is opening a lot of doors to actual application to solve real ag issues quickly, which is really exciting, I think. We're like in an exciting time in technology.
Yeah, absolutely. Let's come back to your downy mildew work. You said there was these two distinct clades, right, in the cucurbit downy mildew?
Yes.
There were hints of it early on, but it wasn't until you actually sequenced full genomes that you realized that there were these differences, or were like some of those differences playing out at like using the standard housekeeping genes and things like that, or?
That's a good question, and we have done both. So we did some whole genome sequencing with Illumina back when. That was kind of like the top technology, right? And there were differences there. We also did like old school microsatellite population genetics, but really high numbers of isolates, 500 isolates are changed. It was very clear there. Right now, we're actually working on a project to try to do high quality long read genome assemblies, but much to my frustration, it is exceptionally hard to assemble correctly a downy mildew genome. Like, I have very strong training in genomics from Sophie and Robin Beale's lab, and it has taken me 12 years to get good quality assemblies from those pathogens.
That's
It
some
has
patience you got there.
it hasn't been from lack of trying. I know it's really funny. So Dr. Rachel Mitchell-Moore is another really well-known downy mildew researcher out of the West Coast, and him and I sometimes would coincide in conferences. And the running joke that he always said to me is, "Don't worry, Lina. It took me like 30 years to get the lettuce downy mildew genome. You're just 12 years. You're doing great."
It's funny.
He was very encouraging always, but it is really difficult. Richard studied several downy mildews, and he's right. It really took him years to get the assemblies to a high quality point, and that has been our experience too with cucurbit downy mildew.
Has like transcriptomics helped to better assemble and annotate those genomes, or is that not something that's used?
I think definitely with the annotation it can help. I think what is hard is the assembly because there's a lot of inherent contaminants in a downy mildew sample. There's really no way for you to grow it sterile. So when you assemble those genomes, you end up having all this like chimeric contigs that have like insect chunks, fungal chunks, plant chunks. And because the databases like GenBank, they're excellent, but they don't have everything that's out there perfectly annotated.
You're only as good as your database.
Exactly. You're only as good as your database. So then you go and compare and you end up with a lot of data that you just don't know. Is it really downy or is it not downy? And again, the long read data is definitely helping. I think clarify some of that, but even with that is difficult. So it definitely has taken a while, but we're getting closer.
And I think it's a good reminder that you brought up is that when you're working with obligate biotrophs that can't be cultured, so these can't be cultured, so you're basically like sequencing metagenomes.
Yes.
And then you have to bin out the individual genomes and not deal with all the noise of that, plus other complicating factors. Yeah.
It's a really complex issue that I honestly was very naive about at the beginning because I had never dealt with a sample like that. So in genomics, you have this very specific kind of calculations that you make of how much sequencing do you need to do to get coverage of this many times of a genome this size, right? So I used that, and every time we did, we would of course come up short because 70% of the sample were contaminants and 30% of the sample was the actual downy mildew, so we were coming way low. So I think once PacBio data got better, we made a really big investment in sequencing a lot of data. Like we probably really overkilled it, like massively. But honestly, that's what was needed. Like we needed to do 300 times what we were doing before because the data just gets eaten up by the contaminants.
Yeah, you want that good depth of coverage, right?
Yeah.
Okay, thank you for that. Now, you've mentored numerous early career scientists that really have ended up working in industry. You look at your CV, Corteva, Syngenta, BASF, Bayer. Is this due to the applied nature of your lab's work? and partnerships with those companies? Or is this more of a deliberate effort to keep a pulse on what industry is doing? It struck me as someone who's put a lot of people in industry positions, and I'm aware that we're at a moment where the current loss and the future loss of federal opportunities are just apparent all around us. And people that might not have considered industry before might think, I should probably think about industry. Like, could you talk a little bit about how you train students to make them competitive in those kinds of industry positions, and what advice might you have for someone that's interested in pursuing a job in industry?
That's a really good question. And it's really funny because the answer has a lot of parts. So,
Yeah.
most people actually that have come to my lab have 100% told me, I want to be a faculty member.
Yeah.
This is my dream. And I'm like, okay. And then you're right, because of the work that I have in my lab, we really work very closely with companies. I think that I love North Carolina because it has a very unique agricultural ecosystem that I don't think is common in other places. We have NC State and we have Research Triangle Park just 20 minutes away. And the grower groups in the state are very closely just working with the university. So we have this constant interaction of like consultants, extension agents, grower groups, representatives from seed and chemical companies, and then the university that is happening
Yeah.
when you're working in extension. And I think once the students and the postdocs get exposed to that, they love it because it's very much about science, but it's also very much about community. And I think that really enchants people. So I really have had several people in my lab tell me, I thought I wanted to be a faculty member, but I don't want to leave North Carolina. This place is great.
Yeah.
It's a great agricultural ecosystem. It's an amazing place to live. Let's talk about other career opportunities that would allow me to continue to be part of this. So then we have the conversation and I tell them like, oh my gosh, there's so many. You could be in APHIS. You could be in one of the companies. You could be in IR4. You could even be working with some of the grower groups. I have some of that. So my technician that retired years ago left to work in industry at first, but now he's actually working with Mount Olive Pickles, which is one of my really big stakeholders. So it's great because if something is happening in pickles and peppers, I hear it first than anybody because he calls me and is, Lina, oh my gosh, you won't believe this thing that's happening. And then another student is the seed rep for vegetables in Bayer. Same thing, I work with him with text every other week. Another postdoc went to BASF. He has a really excellent position as fungicide lead, and he helps me sometimes when I have extension emergencies, like figure out like, is there a BASF product that could help you? And same things with Syngenta. It's a very nice community for ag work, and I think people fall in love with it. And even some folks have left and then come back. Have, for example, gotten a job at Corteva, I don't know, in Indiana or whatever, and then come back. That said, I will admit that I love interacting with my students and my postdocs, and I have always been a little bit sad that none of them went to faculty positions because I wanted to be able to work with them, do grants together and these things. And then recently I did have a couple that ended up going in there. So Camilo Parada is the new field crops pathologist at Cornell. So I'm hoping to collaborate there. And then Alamgir Rahman, he started as the new vegetable pathologist in Clemson.
Oh, wow. That's great.
So,
It's funny because you come back to this. I don't want to leave the area. And it sounds clearly North Carolina is doing a lot of things right in terms of the interactions between the land grant and like the industry partners and the growers and things like that. But I think what also appeals to people is they say, not only do I not have to move, but wait, I get paid more? Because as much as the academic life has this flexibility, when you finally start to look at the salaries, you're like, wait, I have to work way harder for less money.
And you're right about that. I definitely have had some postdocs that have just immediately noticed that. And again, I think it's such a personal decision, right? Because everybody is wired differently. And some of them have loved industry and have stayed. Some have gone to industry and then changed their mind and gone back to academia. But I love that many of them have stayed in areas that I can keep interacting with them. That makes it really fun.
We're out of time here, Lina, but I've really enjoyed our talk today, and I really appreciate you joining us on Plantopia to talk about your career.
Oh, same, Matt. This was really lovely. Thank you so much for inviting me.
We've just heard from Dr. Lina Quesada, William Neal Reynolds Distinguished Professor of Vegetable Pathology in the Department of Entomology and Plant Pathology at the North Carolina State University in Raleigh, North Carolina. As with all our episodes, be sure to check out the show notes for links to Dr. Quesada's research program. I'm Matt Kasson, host of Plantopia. Thanks for listening.